Good morning. My name is Wim van der Poel, and I'm asked to present about the global One Health approach on zoonotic transmission of SARS coronavirus 2. This picture shows where I'm based. The top picture is the Wageningen University campus. And on the lower picture, you can see the Wageningen Bioveterinary Research Site in Lelystad. We have a site where we have a high containment unit. And in this high containment unit, we can work with dangerous viruses like classical swine fever, foot and mouth disease, and also SARS coronavirus 2. Outline of this presentation, I will start with an introduction on zoonotic viruses, spillover of such pathogens to the human field, the global changes that we face at the moment, and I will focus on two of those important changes, including biodiversity loss and climate change. And then I will focus on the SARS coronavirus case, emerging zoonotic viruses, and the global One Health approach we need to cope with that outbreak. And I'll finalize by discussing preparedness for future epidemics. Zoonoses are very important. There is an increasing number of zoonoses that we are facing. About 7% of all new emerging infections are zoonoses. So these zoonoses are very important for public health, and we have a number of animal species where these zoonoses come from. There are different stages of zoonotic adaptation of viruses to humans. For example, if you look at rabies, there is only direct transmission from animals to humans. But when you look at more adapted viruses like HIV, you see full adaptation to the human field. So this virus originates from monkeys, but has fully adapted to the human field. This is also comparable for SARS coronavirus 2, where we see a large outbreak in humans, and the role of animals in the epidemiology it's rather small till now, but that may change when more animals become susceptible to the virus. There are a lot of global changes, and these global changes drive emerging infectious diseases. On this picture, you can see a picture of global warming, changing vector abundance. When there are more vectors, there is more potential transmission by vectors of zoonotic agents. We have a growing world population. That means that we have a lot of susceptible people to potential zoonotically transmitted viruses. We have intensive animal production because we need more and more food to feed the world population. And these animals can be susceptible to zoonotic agents and transmit that to people. We have an increased travel and transport. That means that infected animals or infected people can travel to other parts of the world in a very short time and potentially be introducing new agents in other regions. Changing land use is also a risk factor for emerging infectious diseases, because changing land use means that we have movements of animals and susceptible species for the different ages, and also different or changing contact structures between animal species, which means that we may have species jumps of zoonotic agents. Well, focusing on a few of those risk factors for emerging infectious diseases. First, the loss of species, the loss of biodiversity. This picture shows that in a number of a large group of species like birds, mammals, amphibians, we have an increasing number of species, a loss of biodiversity. Biodiversity loss can be a risk factor for emerging zoonotic agents. Biodiversity loss has different um, ways of influencing the transmission of agents between species. You could say that an increasing number of species could house more pathogens, but on the other side, uh, there is also changing contact structures between species, which means that there will be a changing transmission of agents between species. In general, we assume that these changing contact structures between different species have an effect on transmission of agents between species and also on the emergence of zoonotic agents in humans. Another important risk factor for emerging zoonotic agents is global warming. In this picture, you can see the melting polar ice, the Arctic greening, and drought as a result of global warming. And this global warming, for example, this is a picture of the West Nile virus infections in Europe, has resulted in a changing abundance of vectors, the vectors that also transmit West Nile virus. And in 2020, we have seen the first emergence of West Nile virus in the Netherlands. West Nile virus is moving northward due to incursion of new vector species in regions where we did not have these vectors before. Now to the COVID-19 pandemic. This picture shows a number of photos of what we have seen in the pandemic. The reservoir species, the bed, the wet market where first cases of COVID-19 were seen, severe infections and overcrowding of IC units in hospitals. There was discussion about the work of the Wuhan Institute of Virology, and on the lower part of the slide you can see the sampling and also the protection that we are now using, the personal protection, to try to prevent from spreading the virus. Well, what was the origin of SARS coronavirus 2? It's clear that the ancestor of the virus was in the Chinese horseshoe bat, but it is still a question how that virus was transmitted from that Chinese horseshoe bat to humans. The most likely explanation is the fact, or is the potential presence of intermediate hosts. Another option is direct transmission from the horseshoe bat to humans by a change of the virus. And there are also people that assume that the virus may have escaped from a lab, but there was never found any evidence for that lab escape. So the most likely explanation till now is the intermediate host that has transmitted the virus from the bats to the humans. First cases of SARS coronavirus were found in a wet market in Wuhan. And this picture shows a few pictures of that wet market. And in environmental samples of that wet market, more than 300 samples, 33 were found positive, and 31 of those 33 were from the wildlife trade area. So that made it very likely that there was the virus was coming from a wildlife species, although that was never proven. What kind of animals were traded in that market? Well, in that market there were a lot of different animal species traded. This is just a subset of those animals, and it's very well possible that in between those species there was one susceptible species or that could have acted as an intermediate host for SARS coronavirus 2. What could have been that intermediate host? There is still no definite proof for that. But for example, in the pangolin, the coronavirus is circulating in the pangolin. We see parts of a genome that we also find in the SARS coronavirus 2 virus. So the pangolin is seen as a potential candidate to have been the intermediate host. After all the discussions about researchers have gone into a lot of research to try to find that intermediate host. And for example, this pangolin could have been one of them, also because the pangolin lives in regions where we also see that Chinese horseshoe bat, and that is shown in this picture. The green is the green colored area is where the pangolins live, and the blue dashed area is the area where the Chinese horseshoe bat lives. So there could have been contact between those species. This is a table of a number of species that were tested to be susceptible for SARS coronavirus 2. And what you, for example, can see here, that ferrets and mink show clinical signs, show virus replication, and also excrete the virus. And on the other end, we also see birds and poultry that have never been proven to be susceptible to SARS coronavirus 2. But there is an increasing number of species that is shown to be susceptible and that also seem to be able to transmit the virus. For example, we ourselves have done studies in cats, and in this slide there is a picture of the pathology we see in cats and also the clinical signs that we see in cats and the virus excretion that we see in cats. So cats are susceptible, and cats can also excrete the virus and potentially play a role in the epidemiology of SARS coronavirus 2. Reports that SARS coronavirus 2 can also be found in wild-tailed deer. This is a recently published paper from the north of America, and you can see in this picture the areas where the wild-tail were sampled, and in the right you can see the positive antibody response in the samples obtained in 2021, and this is very indicative for infections of SARS coronavirus 2 in wild-tailed deer. In the Netherlands, we had an outbreak of SARS coronavirus 2 in mink farms, and this is a slide showing what happened. In the south of the Netherlands, there's an area where there were a lot of mink farms, and in that area, more than 60 mink farms were infected with SARS coronavirus 2. Fatality in those farms was not very high, but there was a rapid spread of virus, and this virus appeared to be also infectious for people. In our research, we sequenced a lot of samples from those farms, and all these sequences could be brought down to 5 different clusters. So it's assumed that there were 5 primary introductions by people in the farms, and subsequently the virus was spread within the farm and also to a lot of other farms. In the first 16 farms, we tested a lot of keepers in the farms, and of these people working in the farms, 97 were tested by either PCR or serology, and 66 of those people, so 68%, were found to be infected with SARS coronavirus 2. And when we sequenced 7 samples, we were able to obtain 7 full sequences of people, and all these sequences were related to the mink sequences. So what happened? The virus was first spread by people into the farms, and then through the enormous excretion of virus by the mink, the people that worked in the farms were infected from the mink. So the infection first went from people to mink and then backwards to people. And this paper was published in Science last year. Well, when we look at zoonosis or risks of zoonosis, we can identify hotspots of zoonosis in the world. And this is published in several papers, especially areas where there are a lot of people, where there is a lot of contact with wildlife and livestock, and also between wildlife and livestock, you see hotspots areas where zoonotic infections emerge. And China and India are such hotspots, but also certain regions in Africa and also highly dense livestock production areas in Europe are hotspots for emerging zoonosis. What can we do about those zoonoses? What can we do to be better prepared or better control zoonosis? It's our belief that we need a global One Health approach to control zoonosis and to be better prepared. And in this picture, you can see people that have worked on the One Health approach. On the left, Robert Furkov, the founder of comparative medicine. Calvin Swaby, the person that coined a veterinarian, that coined the One Health theme, and Jeffrey Copeland, who coined the Global Health team, One Health working together of veterinary medicine and human medicine, and Global Health putting more focus on prevention and also on better healthcare in developing countries. In Wageningen, our university, we coined the so-called Global One Health approach, the combined effort of all the health-related disciplines to improve the health of people, animals, and plants in sustainable ecosystems using an integrated systems approach. How does it work in practice? When you have a pathogen circulating in wildlife, there's a risk of spillover to livestock species that may be with or without vectors. And from wildlife as well as from livestock, you can have a spillover to the human population. And when that happens, and there is spread between people, there may be a pandemic in the human population, and that, of course, we would like to prevent. And what can we do to do that? Try to forecast what pathogens are occurring in wildlife and are occurring in livestock species. Implement control operations in livestock species, a rapid response in wildlife species, livestock species, and also the human population, using different control strategies. And when we are able to do that, we can minimize the outbreaks that we see in the human population and also reduce what happens in the livestock population. What's clear from this picture that we need a cooperation between veterinary health, human health, mental health to make this possible. And that's what we call the so-called Global One Health approach. Pandemics are not new, but we seem to have them more frequent and they seem to become more extensive. So there is more and more reason to be better prepared for new pandemics In this picture, you can see the Spanish flu, the HIV pandemic, and now also the COVID-19 pandemic. These pandemics have followed more rapidly after each other, and have also shown that they are more extensive than the earlier pandemics. So it's very important to be better prepared for pandemics. The WHO has recognized that and has started a program to try to cope with a disease X, a disease we don't know yet, but may cause a future pandemic. And SARS coronavirus 2 was the first example. We have started a program to better control SARS coronavirus 2 in the European Joint Program for One Health, formed a network for SARS coronavirus characterization, better preparedness. And in this network, we hope to be able to better control future coronaviruses that may occur. In Wageningen, we have started a program that's called ERASE, also focusing, this is a long year, a multiple year project, working on preparedness, response, prevention, and recovering and learning about emerging zoonosis. We are putting a lot of research programs under this theme, and we hope to be able to do more research on emerging zoonosis in order to be better prepared in future. And that's where I would like to end this presentation, and I will be happy to discuss this presentation in the discussion session of this conference. Thank you very much.